Hey everyone, how's it going? Good morning. Yes, I'm still here in Michigan. Let's see, yesterday was the first time in, I don't know, 5 weeks, 6 weeks, since something like that, since it was in the 30s, because it's been, because it's been in like the 10s and the and the 20s. So we're in the 30s now. It's like a heat wave. Hope everyone's doing okay. Let me pull up what we need to look at. All right, yeah, that looks right. Cool. Let me share that. Let me share. Okay, boom, share. All right, and then we'll fix this. Okay, good. Make this up in the corner here. All right, make that a little bigger. There's a lot involved in all this, virtual teaching stuff that getting better at it now, but it's definitely been a learning curve. All right, let's go ahead and get started. Hopefully you guys can see the PowerPoint slide. I'm gonna pull it up. All right, good. Like that. Great. All right, so today we're gonna finish the ferns. We did most of the ferns last time, but we have a little bit of ferns left to do in this phylum, Polypodiophyta, the ferns. Before we jump into that, I want to say I've graded the third assignment, and you should be able to pull up the correct answers and stuff now. That should be open to you. So make sure you take a look at that later today. These are the assignment grades so far for this course. Obviously, I didn't include the documentary assignment here because documentary assignment should have been a 100 basically, because it was just watching a documentary, answering the questions. These are the actual assignment questions, right? The ones that we have that are on the different lectures. So as you can see, it's like I said in the beginning of this course, right? These assignments are not meant to be hard. The majority of students are getting A's on this assignment. In all three assignments, right? The first assignment, which is on the left in green, 23 students out of 38 got A's. In the second assignment, just make sure, I heard a noise, want to make sure that everyone's muted. Yes, good. The second assignment, 11 out of 38 students got A's, 9 got B's, and then in the third assignment, 16 out of 38 students got A's. So these assignments are not meant to be hard, and 10 got B's. So keep that in mind. They're supposed to help you learn the material. Does anyone know how to look back at assignments? What tab? And Brittany says, go to grades and click on the assignment. Thank you, Brittany. Yeah, this is something that is always a question of how to do this, because from my end on Blackboard, everything looks different than what you guys see on Blackboard. So yeah, just to reiterate that, if you want to pull up your past assignments and look at what the questions were, what responses there were available to you, what you answered, and what you got right and what you got wrong, go to grades. Go to grades and click on the assignment. And then once you go to grades and click on the assignment, Hira is saying you need to click the number grade you got. Click the number grade you got, and that shall pull. Yay. And Catherine says, thank you so much. Yeah, so that should pull it up. Okay, so that's how you get to see your grades and how you did. But the point is of this slide, most students are doing well on the assignments. Please keep that up. The assignments are not meant to be hard. They are meant to help you digest the information in little pieces over the course of the semester, because this course covers so much material. You cannot cram. You can try to cram, but you cannot cram. It's just impossible. So I've taught this class, this is my, I don't know, third or fourth year teaching this class, and assignments help students do better on exams. And most of the time, your average assignment grade is usually your first, is usually basically very close to what you'll get on the first exam. So please keep that up. If you're doing well, students, keep it up. Students who are getting C's, oh, you're so close, you're almost there. Just like a little bit more effort, maybe looking at the material a little bit longer before taking those assignments, right? Just studying it a little bit more. You're so close, so don't give up, keep going. And then for students getting D's and F's, well, for some of these indicate missed assignments, like students didn't turn in assignments, which you got to turn in the assignments, guys, please. But if you're getting D's and F's on the assignments, my guess is that, one, you're probably not attending the live lectures. You're probably watching the lectures, the recorded lectures, which is fine, but it doesn't give you a chance to ask any questions. Or two, you're tuning into the Zoom lecture, but it's just on and you're not watching it. And it's just like you're, whatever, doing something else, and the lecture's on, but you're not really engaging with it. So if you're getting D's and F's on this assignment, you really need to step it up and work a little harder. Okay, why do I say all this? Because our first exam is a week from today. Our first exam is 1 week from today. You should have gotten an email earlier today that a study guide is now available on Blackboard. So I give you the study guide 1 week, 1 whole week before you have the exam. That's nice. Some professors don't even give study guides, which is like, what is with that? But anyways, yeah, so it's there. My advice, well, we'll talk about it a little bit later today, but yeah, so please pull it up, please read it, please study, please use it as a study guide, right, to guide your study. A question in the chat box. Professor, I tried looking at the study guide and there was no content on it. Well, that's not very helpful, is it? Did anyone else try pulling up the study guide, and were you able to see? Well, why don't if somebody could right now? All right, so other people said that they were able to see it. So what I say to, who was that? Swata, I think I'm pronouncing your name correctly, Swata. Try pulling it up again. If you're not able to pull it up, email me later today and I'll just send you a copy directly. All right. All right. Yeah, and so just keep in mind, anything on the study guide is fair game for the exam. So if it's on study guide, it's on the exam. One thing I should clarify too for the exam. Okay, cool. Swata says, I tried it again and I can see it now. Thanks, guys. Cool, good. I think last time so I want to clarify something about the exam, because I think I got my hairs crossed last time. How's the exam going to work? Well, so it's not going to be quite like I described last time. I kind of got my hairs crossed. Basically, we're not going to meet for lecture when the exam occurs. We're not going to meet. Instead, you guys are going to have your exam. So the exam will occur during our normally scheduled class time, right, on March 2nd. That way I can be available to answer any questions or stuff like that. I think originally I had said something like, it's going to be a block of time, but that I looked back to how it went virtual last time and I realized, no, if it's that way, then I can't help you guys along during the exam. So it'll be during lecture on March 2nd is when we'll have the exam, but we won't meet for lecture. You'll just go on Blackboard and take the exam, but that way I'll be available during that time in case you run into any issues or problems, and you can just email me. Okay, enough of that. Enough of that stuff. Last time I talked about the ferns, and today we're going to talk about the ferns again. Not the same stuff though, different stuff. We're going to highlight certain fern genera, and then we're going to have a general discussion about, like, how do you study? Like, how are you studying? What do you do? So We're going to talk about that later today too. Perfect timing for in anticipation of our first exam. So these are ferns. Hopefully you should know that by now, even though we only looked at them. Is there backtracking for the exam? The exam will open up as a single object. It won't be like one question at a time. It'll be just open. It'll be the same exact format as all of the assignments. So that's another reason we do the assignments. So it's an exact, however your experience is on the assignments, that's exactly how it'll be on the exam. Yeah, cool. It's another reason we do the assignments, because it makes me know that it's working on your end, troubleshooting and stuff like that. All right, any more questions? Here, I had a checkbox question. Is available only at the time of lecture? Yes, it's going to start at 10 a.m. and go through for the entire class time. Yep. If you have a conflict of some import that it prevents you from attending that class, just you need to contact me and we can discuss that. Okay. Let us move on. These are ferns. Hold on. Enough. I need a nice fern break for a second, just to kind of. I'm in the forest. It's nice. There's not snow all over the ground. It's not 20 degrees. It's like nice spring day. There's ferns all around me. All right. Move. Nice little forest sounds. Okay. All right, so recall from that first lecture way back in January, believe it or not, I can't believe it because we're almost in March. We looked at this, right? This is that biological taxonomy. This is how we organize the way that we call organisms, right? This is the way we organize, the way we call organisms. And one thing we haven't really touched on yet is this thing order. So we know we're in the plant kingdom, right? In the first class, we even learned about the different domains of the biosphere. Every class, pretty much, we talk about phylum, pretty much. And genus and species has shown up almost every class as well. But order is not something that we've really come across yet. Because most things that we talk about, we can either talk about it as a phylum or as a genus or a species, right? So far. But today we'll talk a little bit about order. And what we're going to do is we're talk about these orders of Polypodiophyta. So we're in the ferns. And these three things here are some example orders in the phylum Polypodiophyta. And you might notice the ending ales. So you have Filicales, Marsileales, Salviniales. So that ales in the plant world, that ales shows you that you're talking about an order. Just in case you're like, what's she talking about? Ales, this ending here, ales. Also, it's just I'm several slides in and I haven't annotated yet, so I'm feeling uncomfortable. All right, for the order Filicales, remember these are ferns. All members of Filicales are homosperous. So if you look at this graph on the bottom, this is showing you the entire circle represents all ferns. All ferns. So I'm going to get out of this color. I'm going to I'll go to orange. So this whole circle is the phylum Polypodiophyta, the whole circle. But in there, you can see the majority of the species are in this order Filicales, right? So this entire green section, 80% of all the species of ferns that we know of are in this order Filicales. And if you're in that order Filicales, you are homosperous. So the majority of ferns are homosperous. The majority of ferns are homosperous, right? And then in the blue slice of this pie, that 20%, there's about 22 other orders in that 20% piece of pie right there. Many different orders, whereas Filicales is the bulk of the ferns. And some of the ferns that we already looked at briefly in the previous class are in this. Well, that makes sense because the majority of ferns are in Filicales. But today we're going to highlight a few of them, which will help you also when you guys have lab. So here's an example. Oh, and I should say, this Filicales order, they're all terrestrial species, meaning all of the species that are in the order Filicales for the purposes of this class grow on land. In the blue slice of this pie, where there's 22 other orders, there's a lot of different aquatic species in that piece of pie. But if you're in Filicales, you live on the land. And here you go. Here's an example of one of these species that is in this order Filicales. So this is the common Polypody or the rock Polypody. Either common name is accepted. This is a super common fern in New Jersey, New York area, even here in Michigan. The species name is Polypodium virginianum. Just a reminder, look at that species name, right? It's, in this case, it's italicized, right? I could have also written it as underlined. Either one of those is acceptable. And notice the first word, which is the genus, is always capitalized, and the second word which is the specific epithet, never capitalized, right? But Polypodium virginianum. We learned last time that ferns, well, so I'll ask you, going through evolutionary time in the plants, right? Where are the sporangia located on ferns? Where are the sporangia located on ferns? In the bryophytes, in the mosses, sporangia were located on Aceta. Okay, yep. And then in the liverworts of bryophytes, the sporangia were located as a horn growing up from an intercalary meristem. Then we got to the lycopods. Sporangia were located on leaves known as microphyls. Now we're in the fern. Oh, then we were in the horsetails where the sporangia were located on sporangiophores, which are weird things. And now we're in the ferns. So where are the sporangia located on ferns? And Brittany says sporophylls. Yes, the answer is yes, in that they are located on leaves. They are located on leaves. So that is correct. However, one distinction is what type of leaves do Lycopodium have in terms of their structure, right?
Megaphylls?
Yes, thank you. Yes, microphylls. And what kind of leaves do ferns have? Megaphylls. Yes.
Megaphylls, yeah, megaphylls.
Thank you.
And the other one is micro, yes.
Perfect. Exactly. Thank you. So the point is, yes, in the ferns, the sporangia is located on leaves, but just to be clear, those leaves are also megaphylls, right? They're leaves that have many, many veins. They're much bigger than a microphyll, and they have a different evolutionary history than those microphylls. The reason I'm bringing this up right now is because there's a really nice picture of that here. There's a really nice picture of that here. So let's use pink. Here is a fern leaf, right? Oh, and we learned some of these terms last time. Here is the blade or the blade or the lamina. The green part is the blade or the lamina of that fern leaf. Here's the petiole. Here's the rachis, right? These are terms that we covered last time. But we know, and just got reminded of now, that the sporangia on ferns are located on leaves, in particular megaphylls, and they happen to be located on the bottom of those leaves. So here you go. This is showing you the bottom of a fern leaf from the common Polypody. And do you see all those yellow circles? Well, all those yellow circles are clusters of sporangia. Clusters of sporangia. And this is a harder question, but does anybody remember what those clusters of sporangia are called in the ferns? We only just learned about it last class, so if you don't know, that's okay. But what are those clusters of sporangia called in the ferns? Boom, sori. Thank you, Brittany. Yes, sori. That is a plural term. One sorus many, many sori, right? Sori, not sori. That's how I remember that. Great job, guys. Thank you. Thank you, thank you. Okay, so that's what I wanted to point out of this. You could clearly see those on this species. Big, beautiful yellow sori. Oh, a reminder, there are fern videos on Blackboard for you to watch, the sporangia catapults, right? So pull those up for fun, but also to just so you get to see plants in action, right? Plants in action. Okay, so common polypody. All right, enough about you. Oh, you know, not yet. Sorry. Here it is again. Here it is again. But this is just to show you. It's another common name it has is this rock polypody. That's because, so I said this is a very common species in New Jersey, New York, Connecticut, you know, even Michigan. But it tends to grow like, so if you ever been like on Palisades, you know, where the George Washington Bridge is, and there's like those cliffs. There's like rock cliffs, and you see sometimes plants hanging off of those rock cliffs, and you're like, how could anything even grow on that? Just like this sheer rock face. Well, rock polypody is one of those plants that is all about that type of area. It's like, what, sheer rock face? Me? Yeah. So that's why I'm showing it you here. I'm showing it you here. Okay. Next, sensitive fern. Oh, it's sensitive. Sorry, I'm getting excited. Oh, it's so sensitive. Question, what is the leaf called again and the stem in between them? I think that what you're asking here, Brittany, is, I think you're asking this. This whole thing is called a leaf or frond. The green part of the leaf is called a lamina. I'll write it in the chat box. Lamina or blade. Same thing. The stem, which I just drew there, of the fern leaf or of any leaf we encounter going forward. Let's put it that way. But the stem is called a petiole. There it is in the chat box. And when the stem continues through the green part, through the lamina, we call it a rachis. And that stuff is also in the previous lecture. I think that's what you were asking, Brittany. If that's not what you were asking, happy to. Okay, cool. No problem. All right, let's go back to the sensitive one. Sensitive. This sensitive fern. I'm not 100% sure how. So I'm Onoclea sensibilis, but you can see that sensitive part, right? Sensibilis. It's sensitive. Why is it called sensitive fern? Well, number one, actually the only reason I know, so all the numbers, is that it's very, very sensitive to frost, So if it's very useful actually, because let's say, maybe not useful for everyone, but it can indicate whether or not an area has already experienced a frost. So let's say it's autumn or fall, right? And you're not sure, and you're just curious, like, did this area have frost already? Like, where are we yet? Are we gonna have snow soon? What's the weather like? Well, if you see sensitive fern, and I'll change colors to, I guess, bright blue. If you see sensitive fern and it's this bright yellow color, ultimately turning even a brighter yellow color, it shows that frost has occurred, because as soon as frost occurs, the leaves of sensitive fern, they're very sensitive to frost, and so they immediately start to die and turn yellow. And so it indicates that frost has already occurred. So that's why it gets its name. Another cool thing about sensitive fern is, and we came across this, I think last time, but also in other classes, this plant has what's called leaf dimorphism. So remember, I'll write it in the chat box. Leaf, oops, don't, come on, move this, there you go. Leaf dimorphism. I wrote it in the chat box. Di equals 2, and morph equals forms. two forms. Leaf dimorphism, it's a plant that has two forms of leaves, two forms of leaves. And what does it mean in this case? In this case, it means that there are both photosynthetic or green leaves and other leaves that are not green that's job is only to carry sporangia, whose job, those leaves, is only to carry sporangia. So in this drawing, or not drawing, in these pictures, so like these leaves here are the green forms, right? The photosynthetic forms of the leaves. And then these brown things here, one here, whoops, whoa, one here, and even another little one here, are the not green forms or morphs that bear the sporangia. So it's two forms. Yep, okay, next we have Christmas fern or Polypodiophyta acrostichoides. So for what it's worth, you do not need to memorize the species names of these ferns that I'm showing you. You should at least know the common names, right? So you should know common polypody or rock polypody, you should know sensitive fern, and you should know Christmas fern. But you don't need to know Polypodiophyta acrostichoides, although I do recommend at least one time in your life saying acrostichoides because it's pretty fun to say. I mean, when do you get to say something like that? Acrostichoides. Anyways, at least in English, because a lot of our words are pretty boring. Sorry, English. Okay, so this is Christmas fern. Christmas fern is named Christmas fern. It's named Christmas fern because people tend to use this. So first of all, it is not as sensitive to frost like sensitive fern. Christmas fern is pretty robust. Christmas fern can take some frost. It doesn't care. It's like, give me some frost, that's fine. So it tends to stick around longer into the growing season, and people tend to use it for holiday decorations for whatever reason, probably because it's still green, often getting closer to the holiday season. So that's why it's called Christmas fern. The other reason I like mentioning that it's called Christmas fern is because the way that I remember that this is Christmas fern is because the smallest leaves, which I'm circling here, look like a mitten, right? Here's the little thumb of the mitten. Here's the little, look, it's a mitten. You know, mitten, not a glove, right? A mitten. Look, oh boy, that's a horrible mitten. Here, mitten, little thumb. Okay, well, so much for that. It looks like a mitten, you know, like a mitten, something you'd wear on your hand when it's cold. We can kind of refresh ourselves again on some of these terms that we've already covered, right? Here is one whole leaf, right? Or a frond. The green part is the lamina or the blade. We can't really see the stems on the bottom, but trust me, they're there. And there we can draw one. Here, we'll draw one. Yeah, there you go. Petiole. And then the stem that continues through the leaf is the rachis, which I just put in the chat box in the previous. Now, so to remind ourselves of other terms, remember, some leaves are simple, and I'll do my best to draw. Simple, here's a stem. And when you have a simple leaf, remember I told you last time, if you're playing with the stem of the leaf, which I know all of you do, if you're playing with it, then the thing, if it's a simple leaf, there'll only be one thing above it, right? There you go, one thing. But you also have compound leaves. Here's a stem. And if you're dealing with a compound leaf, if you're playing with the compound leaf, there's a lot of things above it, right? So like, here's one thing, here's another thing, here's another thing, whatever, etc. Excuse me. Compound. And so here, the reason I'm pointing this out is because looking at this leaf, remember, this is one leaf here. Is this a simple leaf or a compound leaf? Thank you. All right. Thank you, Brittany. Thank you, Vivian. Yes, compound leaf. Correct. Correct. And because it's a compound leaf, we know that there's parts of it, right? This one is only 1 times pinnately compound, 1 times, because there's only one thing that comes off of each, one division. And each of these divisions, right, because there's only one, we could call it a leaflet. We could call it a leaflet, that's fine. But because we're in the ferns, what's another word we can call it? What's another word we can call that little part of a compound leaf? Little part of a compound leaf. Okay, Benjamin says pinna. Brittany says pinule. Vivian says megaphyll. Okay, so not megaphyll, because megaphyll, and Brittany says, oh, pinna. Yeah, so megaphyll is the whole leaf, right? The whole leaf is the megaphyll. And in this case, this leaf is a compound leaf. So this megaphyll is a compound leaf. So the whole leaf is a megaphyll. But on that leaf, because it's a compound leaf, there's little things. And in the case of this compound leaf, there's only one division of little things, right? One division of little things, such that it just looks like a lot of little leaves on this one big compound leaf. Because there's only one division, pinna would be the correct thing. So like here is one pinna, here is one pinna, here's another one, another pinna. But Brittany, you weren't completely off, because I also said in the previous class that when you have many levels of pinnately compound, so like if you're 2 times pinnately or 3 times pinnately or 10 times pinnately compound, the smallest thing, the smallest division is a pinnule. So in this case, yes, it is a pinna, but it is also the smallest division. So if for example, if you had written pinnule as a response on an assignment, I would have given you full credit for that. All right, and so I know this is like, these slides are taking forever, but I'm trying to reinforce those terms that we learned in last class. So on this pinna, on all of these pinna, on the underside of the fern leaf, you can see these orangey things on the underside, and those are the sori. Those are the sori. Christmas fern. Okay. Here's Christmas fern in the forest. You can see the undersides of the leaves bearing the sori very clearly. I think this is our last highlighted species. Okay, no, but we just got a couple more, and I'm going to move through them a little faster. Just a bit faster. Okay, this is the maidenhair fern. The main thing, so first of all, there's 2 things about this. One, this is a case, even botanists say, forget it. we're not even going to try. Here, I'll use red. So just so you know, this right here is one leaf. And this is one of those cases in terms of, this is definitely a compound leaf, right? Because if you're holding the stem would have been down here. If you're holding the stem, there's a ton of stuff up there. So it's definitely a compound leaf. But this is one of those cases, even botanists are like, how many times pinnately compound? Who knows? A lot times. Because it's the way that things branch off of these little stems here, it's not super clear. There's like a lot of stuff happening. So even botanists in this case just say it's pinnately compound. How many times? We don't know. We're not even going to try. Maidenhair fern is a very commonly sold house plant. You can see it. I've even seen it for sale in Home Depot and Lowe's, things like that. If you have one at home and it's not doing well, I'm happy to help you troubleshoot that. But anyways, yes, maidenhair fern, another example of a compound leaf. This one also has a thing that we talked about last time as well. You might remember in the last lecture that I talked about umbrellas, right? I wasn't having a Rihanna moment. I was literally telling you about these things called indusium, which are little umbrellas that allow the sporangia to kind of stay protected, right? They're like a little membrane. An indusium. I'll put it in the chat box. This is a term from last class, indusium. So if that isn't ringing a bell, go back to the last lecture and pull that up. So some species of fern have an indusium, which is a special membrane to protect sporangia, special membrane to protect sporangia. Some species don't have an indusium, but they have another feature that serves that same purpose. And that other feature serves that same purpose, indusium, is a false indusium. And in the case of the maidenhair fern, this has false indusia, right? That's the plural. I guess yellow. Here they are. So you see these little things. So here, each of these, I'll go back. With this whole thing here is showing a close-up of a part of this leaf. And each of these things here is one. Here is another one. Are the pinna. And on the pinna, on the underside of the pinna, you see these weird little flaps. Here's a flap. Here's a flap. Here's another one, etc. And all that is the outside part of the pinna or the leaflet has folded over, and it's folding over the sporangia. So it's serving to protect the sporangia by giving them, some cover from the elements. But this is not another membrane. This is just the leaf itself, the leaflet itself, rolling over to protect the sporangia. And that's what's called a false indusium, which we covered last time. Question. That's a close-up of the picture on the top left. Well, it's a close-up of a maidenhair fern leaf. So it's not a close-up of that exact picture. It's just a close-up. So it's a close-up of one of these. Not this exact leaf, but a close-up of one of these. And furthermore, it's the bottom side of that, right? Because it's showing you, remember, the sporangia are always on the bottom. And so it's showing you the bottom of that. Yep. This is great. I love how many questions you're asking, Brittany, because this is how you understand the material. And I encourage everybody to be active like this, because this is your time with me here. This is your time with me here. If it's confusing now and it's not making any sense now, how are you going to, it's like RuPaul. It's like, if you can't love yourself, if you don't understand it now, how are you going to understand it when you're by yourself, right? Later. So this is the time to ask me these questions. So I appreciate you, Brittany, for being brave and asking all these questions. All right, another one. Bracken fern. Okay, not going to go into bracken fern, but this is just another example of a false indusium. On this one, the false indusium is just the edges of the leaf kind of rolling over. On this one, the whole edge of the leaf rolls over in the bracken fern. It's just another example of a false indusium. Okay, we're now going to talk about, so we're still in the phylum Polypodiophyta, right? We're still in the fern phylum. We are still in the order Filicales, right? Or the largest order in the phylum Polypodiophyta. And now we're going to talk about a few species from the same genus, because they all have something in common, but they all have a slight variation on it. So we're going to talk about a couple plants right now that are all in the same genus, the genus Osmunda. The first one we're going to talk about in this genus Osmunda is cinnamon fern. Cinnamon fern is called cinnamon fern because, number one, when it gets frosted, all of the leaves turn this kind of cinnamon or orangey color. So that's one reason. Another reason is because, much like the sensitive fern we saw in a few slides ago, cinnamon fern also has leaf dimorphism. Leaf dimorphism, or two forms, dimorphism, two forms of leaves. And it has, much like we already saw in the sensitive fern, a green photosynthetic leaf and another leaf that only has on it sporangia. And in the case of cinnamon fern, the leaf that has sporangia on it looks kind of like a cinnamon stick. There it is, right? There they are, right? Okay. It's another reason why it's called cinnamon fern. All right, so that's the first member of Osmunda we're going to look at. Here's another Osmunda species, right? A species from the genus Osmunda. This is Osmunda claytonia, or the interrupted fern. This fern also has a type of dimorphism. However, now it's not two separate leaves. It's not two separate leaves. It's actually the pinna, or the leaflets, that show dimorphism. So let me show you what I'm talking about. I guess we'll stick with blue for now. Maybe I'll do a lighter blue so it pops a little bit more. So here is one leaf. Here is one leaf, etc. Here is one whole leaf. And I said that in this interrupted fern, we have dimorphism of the pinna, or the leaflets. And in that, some of the pinna are green and photosynthetic, like shown here. And some of the pinna are just brown and sporangia bearing, as shown here. And the reason why it's called interrupted fern is because the entire leaf of the fern is interrupted by these sporangia bearing pinna. So what do I mean by that? So look, here, I'm going to go up this fern leaf, right? Here I am, I'm going up the fern leaf, and this part's all green. And then up, what do you know? Oh man, what do you know? This whole area of this leaf has only these type of pinna that are bearing sporangia. And then if you keep going up the fern leaf, oh, now you're back to green stuff again. And so we say that the sporangia bearing pinna interrupt. See how it interrupts the leaf? You can see here too. Interrupts the leaf. Here, so the pinna up here, once again, are the green ones. The pinna down here will also be just the green ones. And in between that, they're interrupted by these pinna that just bear sporangia, hence interrupted fern. All right. Here it is in nature. So if you ever came across something like this, you might think that something's happening, like maybe an insect is eating it, or you know, if you saw this whole leaf and then you saw these things halfway through the leaf, you might think, huh, what's happened there? Did it get burned by fire, or like did insects eat it? But nope, those are just the spore-bearing pinna. Okay, interrupted fern. I think this is our last, yep, this is our last highlighted member of the genus Osmunda. This is Osmunda regalis, regalis, you should think regal, regal, like regal, oh geez, regal, regalis equals regal equals royal. And the reason it's royal is like it bears its sporangia leaves like a crown, So let me show you what I'm talking about. This is another example where it's the pinna, or the leaflets, that have dimorphism, kind of like just like we saw in the interrupted fern. However, now on each leaf, you can't really see it because that other thing here, but the green stuff is on the bottom of the leaf. The green leaflets are on the bottom of the leaf, and the leaflets, or the pinna, that bear those sporangia are at the top, like a crown, Sporangia leaflets or pinna are on the top like a crown, hence the regalis, right? Royal fern. wearing its sporangia bearing pinna like a crown. All right. Why, Rebecca? Why are you telling us all this stuff? This is too much stuff. No one cares about this stuff. Well, because I'm just showing you how these three we just looked at, cinnamon fern, interrupted fern, and royal fern, are all in the same genus, Osmunda, and they all have a form of dimorphism, which is good for us to know that term, dimorphism, but all three of them have a different variation on it, Hold on one second on that, Benjamin, 1 second. Let me just show this real quick. So they all three have a different variation of dimorphism. In the cinnamon fern, you have leaf dimorphism, where one leaf is green photosynthetic. Actually, here, I'll use green in this case. One leaf is green or photosynthetic, and the other leaf only bears sporangia and is not green, right? So that has full-on leaf dimorphism. Whereas in the interrupted fern, which is the one in the middle, in this case, the leaflet, it has leaflet or pinna dimorphism, and the leaflets or pinna that bear the sporangia interrupt the leaf or the frond. So here you go. Here's pinna that is green and photosynthetic. Here's pinna that is green and photosynthetic. And the entire leaf, which is this whole thing, is interrupted by these pinna that bear the sporangia. And then on the right is the royal fern, Osmunda regalis. In this case, it's another example of pinna dimorphism, right? Where the green photosynthetic pinna are on the bottom, and the sporangia bearing pinna are on the top like a crown. So this is just to show you that. Benjamin asks, the royal fern was 2 times pinnately compound? Yes, it is. Here, let me show you. Move this chat box. It's actually easier to see on this drawing than it is on the actual fern. But yep, so look, if you're going up the stem, once you get into the rachis, here is the first division, right? So this whole thing is the first division. And then each of these things is the second division. So we would call one of these a pinna, and we'd call each of these, the smallest things, pinnules. Yep, 2 times pinnately compound, correct. All righty. Then that's great way to be thinking, you know, kind of like, okay, let's put this all together. All right, I think we're done with this. Oh, look, aren't I nice? All right, so that was Filicales. Now we'll talk briefly about some other orders in this fern pie. Remember, I said in the blue part of the pie, there's 22 other orders. We're not going to talk about all 22. We're going to talk about two of those, which is Marsileales and Salviniales. Remember that ales ending makes you know in the plants you're in an order. So Marsileales and Salviniales are part of this blue slice. How much of that blue slice are they? Here's that blue slice shown as a circle. And you can see the Marsileales and Salviniales are just a small part of that blue portion, right? Just a small part of that blue portion. Because remember, there's 20 other orders. So yeah. There you go, numbers, right? Marsileales is 3% of that blue slice. Salviniales is 1% of that blue slice. And then all the other 20 orders are 96% of that blue slice. So these aren't very big orders, right? These aren't very big orders. Marsileales, remember I said that Filicales, they're basically terrestrial, or they live on land for the purposes of our course. In Marsileales, they are aquatic. So aquatic means they live in water. We learned this term. I guess I'll use purple. We learned this term emergent in the horsetails. If you're an aquatic plant and you're growing in water and you're emergent, it means you stick up above the water. You emerge from the water. So most of these species in Marsileales are emergent, but sometimes they appear to be floating. But look at this. Here you go. All members of Marsileales are heterosperous, which is different than the Filicales, because in Filicales, the terrestrial ferns, they were all homosperous. Now we're in Marsileales, they're all heterosperous. And I don't care about this so much, but you will see this term in lab, sporocarp. And the sporangia are on these modified leaves, and we call that leaf a sporocarp. I don't really care so much about sporocarp, but you will see that term in lab, just so you don't get confused. Here you go. Here is Marsileales, one example of it. This is Marsilia, from the genus Marsilia. And so that's easy because it's from that same order, same name. And this one is called water clover. And what do you know? This for sure looks like clover, right? Isn't it like St. Patrick's Day is coming up, I guess, soon. But look at this. Doesn't this look like a four-leaf clover, right? That's actually a fern. It's actually a fern. It's floating in the water. How would you know? How would anybody come across this and be like, that's a fern? Rebecca, that's a fern. Well, I admit, I've seen these in real life in the lab, and yeah, no, they don't look like a fern at all. They look like a clover. Like right now, if you go to the grocery store, a lot of places are selling clovers. They look just like that. But there's two ways you could tell. There's two ways that you can tell. There's two ways that you can tell. Number one, well, it has this weird leaf thing, which is the sporocarp thing that showed up on the previous slide, right? So this is a special leaf that bears sporangia. So that's one way you could tell. But even that, it's like, that doesn't look, that could just, if I didn't know any better, that could be like, that's a sick leaf, right? Or that's a leaf that's still growing. I wouldn't know just looking at that, that's a sporocarp or a leaf that bears a spore on it. Like, I wouldn't know that. But here's something, this is a dead giveaway. What the heck is that guy there? What is that? What is that thing? Ah, yes. That, my friends, is a fiddlehead. That, my friends, is a fiddlehead, AKA crozier, right? Sometimes you might see the word crozier. Fiddlehead, AKA crozier. Either is correct, but fiddleheads are something that only ferns have. Only ferns have. So that's why we know this is a fern. It's one of the reasons, but that's the easiest one. Oh, there you go, sporocarp. So that's our example of Marsileales. Now we'll talk about Salviniales. These are also aquatic ferns. Usually they float. And just like the other ones we just looked at right now, Marsileales, in Salviniales, all members are heterosperous as well. So they also have this special leaves that have on it sporangia that I don't really care about the term sporocarp. Here is Salviniales. So you might have remembered in the very last time we talked about ferns, and I talked about the diversity of ferns, and one thing I highlighted was size, because ferns have in their phylum some little teeny tiny itty bitty plants, and then like trees, right? Well, these Salviniales are examples of the little itty bitty plants. Wait, duckweed is the only known plant that has B12. Well, so I didn't know that. I'm guessing you're saying vitamin B12. I will say though, there's actually many different plants that are called duckweed. I know of at least 4. one of which is a fern, which is this one. And then later on in the course, when we get to the flowering plants, there's another plant there that's also called duckweed. So thank you, Catherine. That's a really good example of why it's important to know at least the genus or species name of the stuff you're trying to make vitamins out of, because I don't know which duckweed you're talking about that has B12 in it, but if it's not this fern, you don't want to be walking around in the, and you see this growing this grows in Prospect Park. I mean, you don't want to go there and like, yeah, but the other one, Catherine wrote, I know it grows in water like this. Yeah, so does the other one that we'll talk about though. So all the ones I know of grow in water like this. But yeah, another reason why common names are not necessarily useful when you're going to make things like medicine or vitamin extracts from them, because I know of at least 5 things called duckweed, but I digress. We're talking about this one right now. This one is a fern. Look at that finger on the top here. That finger is holding one whole mature sporophyte plant of this azolla or duckweed. Look how teeny tiny. Look how teeny tiny. Super teeny tiny. Duckweed, in this case, this azolla, forms these dense mats. So this is somebody put their hand in this water and pulled up, and look at all this duckweed that comes up on it. They form these dense mats in water. And actually, we'll get to that in a second. Symbiotic relationship alert. Duckweed has a symbiotic relationship with cyanobacteria, also known as Anabaena. You don't need to know Anabaena, just cyanobacteria is enough. And in this symbiotic relationship, duckweed provides a home for the cyanobacteria, and the cyanobacteria fixes nitrogen for the plant. So on the left here, you can see it, there's a scale. Here, look, 2 millimeters, 500 micrometers. Whoa, crazy, who knows? Oh, here we go, sorry. I was like, what is that? Hold on, undo. That doesn't make sense. There must be a typo. Looks like, I think I made a mistake there. Anyways, from bigger to smaller, don't worry about the numbers. I was trying to give you an idea of scale. Because look, this one is correct. This is one duckweed plant, and look, 2 millimeters scale. So super, super small. Don't worry about the other ones, because apparently they're not good. This is a close-up. Let's say it's from here. And if we were to put this under the microscope and look at the plant underneath the microscope, we would see these guys, right? The cyanobacteria, Anabaena, inside of the leaves for this symbiotic relationship. Here you go. So, and when I say that duckweed or azolla provides a home for this cyanobacteria, it straight up provides a home. There's a whole part of its leaf that is dedicated to home for cyanobacteria. So, wow. It must be very important for this plant to have this symbiotic relationship, or else why would it dedicate such a large portion of its leaf to being a home for these guys, especially when your leaves are so teeny tiny, right? So look, here's one leaf. We're going to look at it closely under the microscope. Here is a cross section through that leaf, and look at that giant leaf cavity full of cyanobacteria. And look how much space that leaf cavity takes up in that leaf. It's a lot of space. Here is a real picture under the microscope. Here's that cavity again. Each of these darker things is the cyanobacteria. And then there's an SEM microscope on the bottom, but don't worry about that. The point is, like, it must be a pretty important symbiotic relationship if you're going to dedicate so much of your leaf space to a house for cyanobacteria. Okay, so that was duckweed, maybe a plant that has vitamin B12, TBD, right? TBD. Now we'll talk about water moss. Water moss is also in the order Salviniales, and just conveniently, we will talk about members in the genus Salvinia. So Another example of a super, super small plant, right? This is the sporophyte we're looking at here. Super, super small, not quite as small as the one we just looked at, but still pretty darn small, right? Still pretty darn small. Here is one plant. Pretty darn small. These also grow in the water. Oh boy. Okay. All right. Let me see. What do I want you to know from here? Okay. So what I want you to know from here, over evolutionary time, this plant grows in water. This plant grows in water and floats and floats. So number one, it doesn't need to be attached to anything. And like, it doesn't need to be anchored to anything. It's floating. And number two, it's in water. It's floating in water. So because of this, over evolutionary time, we'll change colors. This plant has lost its roots. It doesn't need roots anymore. It doesn't need to use resources to make roots anymore. It doesn't serve any purpose. What it does have remaining is a stem and leaves. Here, I'll do this, is the stem right here in blue, and leaves, which I will draw in pink. Here is one leaf. Here is one leaf. And believe it or not, you're not gonna believe it. But believe it or not, this is actually a third leaf. What do you mean, Rebecca? Well, I mean, this is another example of leaf dimorphism, dimorphic leaves, in that this has two leaves that are green and floating. And then the third leaf, which hangs into the water, which hangs down into the water, has lost the lamina and only the veins remain, right? So the green part has been lost because number one, it's hanging in the water. So it's not getting exposed to sunlight. So it doesn't need all that green lamina anymore. So the lamina has been lost, and it serves as roots in the sense that it's the bottom part of the plant that doesn't have any like green stuff on it, but it's not a root. And just so you know, these little, oops, sorry, go back. I should have done this. Lamina lost over evolutionary time, right? Here's that part, lamina has been lost over evolutionary time. Serves as the roots in that they are just the thread-like stuff at the bottom of the plant. But it's floating in water, so keep that in mind. And then the sporangia are going to be on this thread-like part on the bottom, this leaf on the bottom that no longer has the lamina. Here are the sporangia. Okay, whatever. That will show up in lab. That's another reason why we're talking about it. Oh, look at that. Okay, talking about Salvinia, you may have noticed in the previous pictures, maybe in this one, but maybe not. These are super fuzzy. Like if you were to look at them up close and hold them in your hand, they almost feel like felt. They're super, super fuzzy. And that's because they're covered in trichomes. Trichomes are little hairs that come off of leaves. Sometimes they come off of stems, but they're basically hairs growing on plants. And in the case of this water moss, you can see some examples of the trichomes over here. See each of these things here, these are trichomes, each of these little hairs. And what they do in the water moss, what these trichomes do in the water moss is bubbles get stuck to them. So here's a giant bubble, an air bubble, and it makes it so the plants can't sink in the water. So you can try. I mean, if you ever come across this, you can try push it down so that it sinks in the water, but the hairs get covered in bubbles and they just float right back up to the surface. Both of these plants we just looked at, Azolla and Salviniales, Salvinia, which are genera in this order, Salviniales, are problematic invasive species. What do I mean by that? Look at this. So this was a lake. but the surface of the lake now is covered with one of these, probably Salvinia, covered. And this is a problem. This is a problem for this lake. Why? Number one, it's not a problem for the plant, the Salvinia, right? Because, which one? Probably Salvinia. It's not a problem for that because it's getting water, it's getting sun, it's doing its thing. But think of all the fish that are living in this lake. Think of all the plants that are living in the water now being shaded out. Think of all the nutrients that these plants are putting into that water. It's just not good. It's not good. And eventually it'll make this lake be not habitable by other organisms. So in certain parts of the US, you'll actually see signs warning you to be careful about moving these plants. It's actually illegal to possess these in certain places because of things like this. Oops, because of things like this. Yeah, problem. Here is one way that people have come up with to deal with this. So here you see a boat, and on the left you see a boat, and it's actually pulling behind it this floating rope, a floating rope. And they'll actually collect all the floating plants and then use some chemical to kill it, or manually scoop it out of the water to try to remove it. Questions. Does this called eutrophication, or is that limited to algae? It does. So eutrophication is a process where it's a super common process, unfortunately, in our freshwater bodies in the United States. But basically what happens in that process is so much nitrogen, I think, is it nitrogen? But so much, yeah, I think it's nitrogen. Yeah, it's nitrogen because the plants themselves can contain nitrogen, and as they decompose and all that, yeah. So the freshwater body becomes so high in nitrogen that nothing else can really grow in it, and it takes on this kind of like, as Catherine mentioned, like a green color, because the nitrogen promotes algae growth because it's like fertilizer for algae, and then algae just like goes crazy, and then the algae uses all the oxygen in the process of respiration. We haven't gotten there yet, but the point is, yes, it's bad. And yes, these floating ferns are also in that same category, in that if you have, let's say you have a freshwater lake, and let's say somebody accidentally released some salvinia into that freshwater lake, and then let's say there's a farm next door that is using fertilizer, and the fertilizer goes into that freshwater lake, and now that freshwater lake has a lot of nitrogen in it. Salvinia will be like the first thing to kind of take advantage of that, just like that algae, and you will end up with something like this. So yes, great way to put that together ecologically. Okay, we're almost done here. Are we? I hope not. I hope we are. We got 10 minutes. Another way people try to control this is by using these bugs called weevils that will eat the salvinia. Okay, got about 10 minutes left. And what we're going to use that time to do is to talk about studying. Why are we going to talk about that? Because the first lecture exam is 1 week from today. 1 week from today. So the reason I have this included as part of my lecture is because when I was in undergrad, long ago, not that long ago, I realized that undergrad programs do a crappy job at teaching you how to study. It's almost like you show up to school, you paid your tuition, you bought your books, you show up to class, and they're like, here's information, good luck studying it. I'm going to test you on it next week. That's not fair. That's not right. How do I study? Give me some studying tips, undergrad. So I was always very frustrated by that. And instead, I kind of had to troubleshoot what worked best for me and like asking people what they did, and it just wasn't useful. So I hope one day that in undergrad, they'll have a class that's dedicated to studying. I know sometimes there's resources in like learning centers and things like that, but still, they should focus on it more. So because they don't, I will, a little bit, just a little bit. I will. And I hope what I'm about to talk about will help you not only in this class, but in other classes and in studying for things like MCATs or whatever you're studying for, GREs. Or if you're not going to study anymore, then maybe learning how to do crafts. I don't know. Okay. So how do you study for exams? This is something I would always ask. I always ask this to my students, like, how do you study for an exam? Like picture you studying for an exam or for an assignment or quiz for that matter. And how do you do it? Like, what do you see when you picture yourself? Are you seeing yourself sitting down and like looking at notes and reading them? Like, is that what you're doing? You're just like sitting and looking, right? So basically you're sitting and staring at whatever it is. Maybe you're sitting and staring at notes that you wrote, right? You're looking at notes that you wrote or the book. Or maybe you're staring at diagrams that they're already filled in. They're already filled in with the correct answers and you're just there staring. And you're just hoping, right? Ooh, if I stare at it long enough, it'll just go in my brain, right? So when I ask students how they study, this is what they tend to say, that they do this. And that might work for some students. And if that is what you're doing and you're doing well in your courses, then fine, keep doing that. But this did not work for me. This did not work for me as much as I wanted it to. It just didn't work. So here's why that doesn't work. Here is why. And I'm going to give you an example that has nothing to do with plants. So let's say you want to make a brand new recipe that you've never made before, right? And I'm assuming most of you in here probably haven't made collage, but maybe you have. But let's say I'm like, hey guys, let's make some collage, right? Let's make some collage. And I give you the recipe and I give you the ingredients and you go home and you stare at this for like, I don't know, 20 minutes, right? You stare at the list of ingredients, you stare at the directions for 20 minutes. And then I take away the instructions and I take away the recipe and I say, okay, good, go to the store, buy all the ingredients that you need. And then when you get back from the store, make me collage. But no, you can't have the recipe anymore. That's gone. Collage is good, Brittany. It's like a cookie kind of thing. Anyway, so yeah, like chances are if I'm like, all right, you got this? Okay, great. Oh, chances are, number one, you're not going to be able to buy the right things at the grocery store. That's number one. Number two, let's say you do somehow buy the right things at the grocery store. When you get back, you're not going to remember how to make collage. You never made collage before. This is like a new thing. So reading and staring at the material is not a good way to get it to stick. This is my good example of why. You're not going to be able to make collage. You just stare at that recipe for 20 minutes. Some of you might be able to, and if you are that person, good on you. But I'm not that person. So this didn't work for me. So I'll show you what worked for me during undergrad and also grad school. Better ways to study. That leads not only to higher grades, but longer retention of the material, There's some ways that you've probably heard of before. For example, to study in pairs or small groups. The reason why this is good is because instead of staring and reading, it forces you to kind of recall and explain in real time. So you could do this even, let's say you have a partner or a sibling who also happens to be in school, or a younger child who can read and who can like be, help you with this, or a college friend. There's lots of people who can help you with this. But the point is, give them something to read off of and have them say to you, okay, what is a sporophyte plant? And then you're like, oh man, what is, it's, I don't know, right? So it forces you You know, if they're like, what does monoecious mean? You're like, ah. So it forces you to recall and explain. And that works way better than just reading and staring. And you could do this in Zoom. You could do this on FaceTime. You could do this in a lot of different ways virtually. If you want me to connect you with somebody in the class to have a small study group, let me know. Send me an email. I'm happy to do that. But if maybe you don't have time to do this, or maybe you are antisocial or introverted and you just don't want to interact with other people that much, that's okay too. Nothing wrong with that. So I designed, not really, but this kind of quiz yourself technique, which serves that same purpose of recall and explain, but you can do it by yourself. Here's what you do. These are the steps. You're going to open a Word document. You're going to change the orientation to landscape. These are kind of like, I call them updated flashcards, because they're useful in both a printed format. You could print this out, but you could also download it as a PDF and look at it on your phone. So you open a Word document, you change the orientation to landscape, and you insert a table into that document that has 2 columns and lots and lots and lots of rows. And I'll show you an example on the subsequent slide. You put questions, unlabeled diagrams, pictures from the lab, or a term on the left-hand side. And on the right-hand side, you put the answers or a labeled version of the diagram or definition, etc. Then what you do is you print that out. If you like paper, you print that out and you fold so that you could only see one side at a time. So for example, you fold and you have the questions, you read the question and you're like, I think the answer is this. Turn it over. Yes, I'm right. Or nope, gotta study that more. But the nice thing about making this on a Word doc is you could also save it as a PDF and then view it on your smartphone. So like I used to commute on the subway, and it's not useful to have all these cards, these flashcards. And sometimes not even that useful to have like a printed out document because it's crowded and I'm trying not to fall, and like I'm just surrounded by lots of people commuting. So I downloaded it as a PDF on my smartphone, and then you could zoom in to the left-hand side that has the questions, think about it, and then move over and look at their answer. So here's what it might look like. This is an example, right? You put the question on the left or an unlabeled diagram. So this way it would be like, you would only look at this part. You would only look at this part and you would ask yourself, what does monoecious mean? And then you would say, I don't know. Oh yeah, you know, sex organs are in one house. A house is a plant. Sex organs are on one plant. And then you look, am I right? Yup. So I know this seems maybe silly to some of you and you're kind of like, why is she doing this? But you would be surprised. In the past, I've taught people this and they've actually thanked me. They said it helped them remember stuff. I use this for orgo, for organic chemistry. I use this for abstract algebra. I use this for Calc 2, all of things. I use this for physics, all of which were science classes that I did not want to take, but I had to in my undergraduate days. And that wasn't stuff that I liked or knew about, but it was stuff that I was able to learn using this method. Okay, so the study guide is on Blackboard. What I would recommend you do now that you have this how do you study kind of thing is use that study guide to generate the questions that are on this side. Use that study guide to guide you. And another thing is you'll be surprised if you actually go through the process of making something like this, you'll find by the time you're done making it, you almost don't need to study it that much. Oh, another thing I should mention is you have the lecture slides. You have labs that are PDFs. Hello. Take screenshots of diagrams. Take screenshots of images and like throw them right in here. Right? And then ask yourself questions. So use technology to your advantage. Okay. All right, well, that's all I have for today. If anyone has any questions, I'm happy to answer them now. And I really want you guys to do well in this first exam. I really do. I really do. So please pull up the study guide today. please at least pull up the study guide today. I recommend taking it in sections. The study guide goes from the beginning of this course and then down, like until the horsetails, which is what the exam goes to. So please pull it up. And if you have any questions about material that's on that study guide, feel free to email me. If you look at your graded assignments, which is another great thing to look at as you prepare for the exam. If you see your graded assignments and you're not sure why you got something wrong, ask me, email me. Maybe I made a mistake, or maybe you're straight up wrong and you just didn't, and nope, you got it wrong. and I'll explain why, But if you're confused about anything, email me. I have office hours this coming Thursday. Someone says, please, how many questions? It's not written yet. It's almost written, but I will say this much. The exam is going to occur during our scheduled lecture time on the 2nd of March. And our scheduled lecture time goes from 10 a.m. to 11:20, right? An hour and 20 minutes. So whatever I do, whatever the exam is, will have to be an exam that can be given in that amount of time. So what I'm doing is I have older exams and I see how long those older exams were that I would do in person. And I'm adapting the number of questions that we would have had in person to be conducive to a virtual exam. The exam will be just like the assignments that you guys do, same format, same types of questions, available to you all at once, not one at a time, so you can go back. Yeah. So if anyone else has any questions, I'm happy to answer here. Otherwise, have a great day, and I will see you guys on Thursday. Thank you. Have a great day.
Have a good day, Professor.
Great day. Have a question here. When will the attendance be updated? I'm planning to finish that by today. It's on my list of things to do today. So I haven't forgotten. It's just grading the assignments took longer than I was expecting, and then I started working on the exam, and I wanted to make sure that you guys had the study guide. So the attendance thing is it's on my to-do list. Thank you. Have a great one.